Today we're going to talk about properties of skeletal muscle. That's hugely review from anatomy, but I recognize it might have been a while since you've had anatomy. So it's warranted to review the structures that we find inside a whole muscle. But we're also going to study how muscle contracts. And you need to be mindful of excitation, contraction, coupling, the processes of contraction, and what's required for relaxation. 4 things. So when I get to that and I go through it many times, pay attention to that. You will have an announcement that posts on Friday telling you what to expect for next Monday, and we're going to be discussing the PhysioXs. I do recommend that you do PhysioX2 numbers, activities 1 through 6. It's not due yet, but that's when I'm going to talk about it, and it will help you, I think, for your lecture exam as well. That's just my little, not warning, just suggestion. That's what it is. So, does anybody recognize the person in this picture? It's Arnold, of course it is. And you know what? I think he still looks pretty good for his age. Don't ever say aging sucks, because only a privileged few get to do it. What's the alternative? Death. So celebrate it. It may not be very graceful, but the more you accept it, the more graceful you do look as you age. The functions of muscle include not just movement, but they also help with heat production. In lecture unit 1, for example, I'll remind you, when your body temperature was too low compared to set point, which might have been higher, we talked about one of the ways to compensate was, bless you, small, minute muscle contraction. That's what we do when we shiver. It is also not just for movement, but the fact that all of you are sitting upright for the most part in your seats. It's important for posture and also for joint stability. Muscles contract to stabilize different joints so that the primary mover can actually move them appropriately. We also are a unique species because our facial muscles actually insert into our skin, and therefore it can pull on our skin and gives us wonderful facial expressions like that one, or smiling, or doing things like winking. Yeah, there's a lot we can convey with our facial muscles. And most of the time we don't even realize that we have an expression that looks like we're disinterested. I mean, I know there's a term for that. I don't want to say that on video. I am 100% guilty of that expression, but I'm not resting in my head. I'm either having a party, highly entertained, or I'm thinking about all the things I'm going to do. I'm thinking ahead. We also, it supports abdominal organs, like our 3 layers, but 4 muscles of our abdominal muscles. They help hold our intestines in place. And also, lastly, it helps control body openings, like your eye. Let's go. Close them. Open. Wink if you can. My ex-husband cannot wink to save the life. He can't. He just can't. He just goes. So that's blinking, not winking. Close your mouth. Pucker your lips. Now pucker other sphincter muscles down below. Go ahead. So it controls body openings. Skeletal muscle, well, all muscle really, is excitable. And we have already learned about resting membrane potential and action potentials. We know that excitable cells can change the resting membrane potential. They can depolarize, they can hyperpolarize, they can depolarize to threshold and spike in action potential. They're excitable. Skeletal muscle is, so is cardiac muscle and smooth muscle. They are excitable tissues. They can do the same thing as a neuron. They also have conductivity, which means they can transmit, they can conduct this electrical impulse from one part of the muscle to the next. Just like an axon for the neuron conducts that electrical signal all the way down the axon. As a result of this, we know that there is going to be a mechanical event. How many times do I need to say this? All mechanical events are preceded by an electrical one. And most of the times, the students think, oh, okay, muscle needs an action potential to contract. And that's true, but it also needs the downward stroke. That's repolarization, I'll remind you, in order to relax. We forget that the process of relaxation is also a mechanical event. They have extensibility. They can be stretched. So you go to yoga class, you stretch your muscles, downward dog, pigeon, happy baby pose, all of that, all those stretches feel amazing. And yet, even though they're extensible, meaning elastic can be stretched, they also have this elasticity, which means they will recoil. And that's great because when you stretch and you leave that yoga studio, you're not dragging your knuckles on the floor, right? The muscle recoils. It has elastic tissue with it. This should be review for you from anatomy. But again, as I said, it might have been a while since you've had anatomy. Some of you might have had it last semester, some of you the semester before, I don't know. So I'm going to hedge my bets and cover this. There are 3 types of muscle. 2 of the 3 are involuntary: smooth muscle and cardiac muscle. 1 is voluntary, skeletal muscle, and they do have characteristics. Skeletal muscle is striated. The fibers, and the word fiber refers to a single skeletal muscle cell, a single cardiac cell, a single smooth muscle cell. Fiber refers to one cell. In our neuro discussions, a fiber referred to a single axon. So the word fiber has multiple uses for us in this unit. Skeletal muscle, to go back to it, is striated. We're going to review why it looks striated, and it comes from, short answer right now, how the actin and myosin, the thin and thick filaments, interdigitate with each other in a regular arrangement so that it looks like it's lined, like lined paper that many of you are writing on. It is multinucleated because, and I'm going to explain that to you on another slide, a lot of the myoblast, the word myoblast means not completely mature. It's still developing. Think blast with B and B for baby. They're like baby cells. And a whole bunch of myoblasts come together and fuse to create one large skeletal muscle cell, and all of them bring their nucleus with them. And the nuclei in skeletal muscle are kind of pushed off to the borders of the cell membrane. Cardiac muscle is also striated. The cardiocytes, muscle cells are branched. You're going to know that and see that when I start lecture unit 3 next week. Can you believe it? I know. But it's okay. At this point, we've had such a good platform. Lecture unit 3 and unit 4, if you study with Caitlin, she would tell you, you got your groove going. Now it's just, which organ system are we in now? You've got your language. So it does get more approachable, let's put it that way. Cardiac muscle not only is branched, but it has unique structures called intercalated discs. Have you ever been in a hotel room where inside the hotel room there was a door, and if you unlocked it and the people next door to you unlocked their side, like if you had family, you could walk back and forth between the hotel rooms without having to exit and use a card key. You don't have to go to the breezeway. Have you seen that? That's how intercalated discs behave. They are modified gap junctions. I want you to think of that internal hotel door on both sides being unlocked because both cells have to bring their gap junctions together and align them. And then you have a thoroughfare, if you will. And that means the electrical current in cardiac muscle can spread from one cell to the next directly. And that's a huge feature. The other thing is cardiocytes, for my testing purposes, are mononucleated. Every once in a while, we might find a cardiocyte that has 2 nuclei. We're not going to entertain that. We're going to say mononucleated, okay? Sure. It is involuntary. And then lastly, we have smooth muscle. It is spindle-shaped. It is mononucleated. The thick and thin filaments are not arranged in an organized pattern. Therefore, it is not striated. And again, it is involuntary. We find smooth muscle lining blood vessels, the middle layer of blood vessels called the tunica media. We will be reviewing that in lecture unit 3. We also find smooth muscle cells lining your digestive tract. We learned about digestion earlier this week and last week. It is lining the intestine to allow for that peristalsis and pendular motion that I was telling you about. So they are involuntary, spindle-shaped. Another word for that is fusiform. Here is just a nice picture from an anatomy book in case you're not looking at yours. At least you have it on your slides. And it highlights these very characteristics that I just told you about. If you see the prefix myo or sarco, you should be thinking about muscle. Muscle mainly gets bigger because when you're working out, the cells you have get bigger. That's called hypertrophy. And here in the picture, I'm showing you many myoblasts that start to fuse to become a skeletal muscle fiber. And when they all fuse, we get one giant fiber with lots of nuclei. Satellite cells are support cells. They help with this development. So you don't make more muscle cells. You merely make the ones you have bigger. Now, when I was in graduate school, I had 2 friends in graduate school. One was actually studying muscle regrowth patterns because we oftentimes believed that skeletal muscle at a certain point is post-mitotic. You can't grow new ones. You can't develop new ones. And for the most part, that is true. But Esther, one of the graduate students, said in her research, and she published this, she actually discovered that the myoblasts still can have mitotic activity. So if a child is injured when they're young, they may grow back some of that skeletal muscle. And I'll give you an example of that. My young cousin when he was 5, they lived out on a farm, they had acreage, and he was on an ATV by himself, age 5, just having the time of his life. And all of his brothers were with him, and he somehow flipped over the ATV and it crushed his biceps brachii. Got a lot of damage from it. He lost a lot of muscle mass from that injury. But he, right now, as an adult, has greatly recovered. The injury happened when he was young. He didn't fully regrow the biceps brachii, but he had some partial growth of it, and he can use his arm just fine. So the studies suggest that if some sort of muscle trauma happens to you when you're really young, and I don't suggest that at all, I'm just saying there could be some partial recovery because there are still some myoblasts. F words. We all love our F words. So now when you get frustrated, I want you to say, oh, fascicles, or a mother fiber. Use one of these F words. Ever notice that it's not the word itself that seems to be offensive, it's the way you say it? I can turn to Rebecca and go, you Kit Kat! And it sounds offensive. It's funny how society is with that. But here are your F words. Fascicles, fibers, fibrils, and filaments. The filaments, I'm going to start very small and then get bigger. Filaments, and let me turn off this front light so you can see things better. There we go. The filaments, or also called myofilaments, because remember that means muscle, consist of thick and thin filaments, and I will go into more detail about the thick and the thin filaments. Getting bigger are fibrils, also known as myofibrils. These are rods of protein, rods. And within them, we have the myofilaments. That's why it looks striated here. These myofibrils are inside the muscle cell. A single muscle cell is called a fiber. And here we see a fiber with the cell membrane removed, the endomysium, which wraps around an individual muscle cell, and coming out like this right here. It looks like someone is taking out a single cigarette from a cigarette pack, right? So think of a single cigarette coming out of a pack representing a myofibril. Here is the entire fiber, one cell. Now, of course, we can have many muscle cells, fibers, clustered together in a group, and this is called a fascicle. If you go to the grocery store, even if you don't eat meat, just walk by the meat department and look carefully at some of the thinner cut steaks, and you will see fascicles. In between each fascicle, we have connective tissue called perimysium, and that creates the marbling effect in a lot of meat. Then, if we have a whole bunch of fascicles grouped together, we actually have a muscle. And there is a wrapping around the muscle called epimysium. It is continuous with the tendon of the muscle. So epimysium around the entire muscle, perimysium around a fascicle, these clusters you're seeing here. And around an individual muscle fiber, we have endomysium. And below it, we have the sarcoplasm. Don't let the word sarcoplasm scare you. It is simply a fancy word for the, let me strike that, back up. Underneath it is the sarcolemma. Don't let that scare you. It sounds like plasmalemma. It just means the cell membrane of the muscle cell. Inside is the cytoplasm, but that's called sarcoplasm. Again, sarco refers to muscle. And we're also going to learn tonight, if you haven't already, about sarcoplasmic reticulum. That sounds a lot like endoplasmic reticulum, doesn't it? Yeah. So don't let those prefixes scare you. You know your language. You've got this. So now we have a single muscle fiber. We've removed the endomysium, and now we have removed the sarcolemma. We're taking out one of these myofibrils, rods of protein. And those myofibrils have myofilaments, the thick and thin filaments. Something else I want you to pay attention to. There's a lot of mitochondria in the sarcoplasm. The nuclei, as you can see here, are pushed against the sarcolemma. Here's another nucleus. Here's another nucleus. They're pushed out to the side to make more room for the myofibrils, these rods of protein. In this kind of blue-purple color, you see the sarcoplasmic reticulum. It is endoplasmic reticulum. And this organelle in skeletal muscle holds a lot of calcium. A lot of calcium. Now, when we were doing our table for neuron physiology, remember I told you that calcium is found most abundantly outside of the cell? Well, there's a lot of calcium inside the cell, a lot, but the trick is, it's not a trick, the caveat is it free? Is it freely available? Inside the cell, organelles like mitochondria or the endoplasmic reticulum can sequester calcium, which means it's not free going around the cytoplasm. When and if and when calcium is allowed to enter the cytoplasm of a cell, especially an excitable cell, great things can happen. Neurons will release neurotransmitter. Muscle cells will contract. Doesn't matter which kind of muscle cell. Universally, all three of them require calcium in order to contract. So then, Kira, if great things happen when the calcium is free in the cytoplasm, why not just let it be free all the time? Too much calcium dumping into the cytoplasm can be toxic to the cell. Do you remember when I talked to you about glutamate and how it has excitotoxicity? And I talked about brain trauma and how the neurons that are traumatized can just vomit their neurotransmitter. If it's glutamate, it opens calcium channels. And when a lot of calcium is free in the cytoplasm, that is a trigger to tell the cell to commit suicide. Yep, that's called apoptosis. So it needs to be sequestered. It needs a hold. It's like jail. Until we're ready for it, it needs to be sequestered. There are swollen endings here of the sarcoplasmic reticulum called terminal cisternae. The word cisternae means like large cavernous sac. And in between the terminal cisternae, on both sides of this green structure, the green structure is called a T-tubule. This is where the cell membrane, the sarcolemma, invaginates and kind of worms its way. It doesn't break into the cytoplasm. Kind of worms its way around the myofibrils. How can I get you to understand that? Have you ever blown a bubble with chewing gum? How do you do that? Think about how you have to do that. You have your gum you're chewing. You flatten it out, don't you? Flatten it out at the roof of your tongue. And then you move it so you can start sticking your tongue a little bit through it, right? To make this little pouch or pocket. And then if you push your tongue too far, it breaks, doesn't it? Then you can't blow your bubble. You got to chew it up and start all over again. So this T-tubule, I want you to think of it as the little pouch that you are making with your tongue in bubble gum when you want to blow a bubble. You don't break through it. It just is continuous. And you know the bigger you can make that pouch, if you've got the right bubble gum, you can make a bigger bubble, right? So this is just an invagination of the cell membrane. It does not break through into the cytoplasm. So on either side of a T-tubule, we have a terminal cisternae and another terminal cisternae. 3 T's in a row. Terminal cisternae, T-tubule, terminal cisternae. 3 T's in a row, we call that a triad. So triad. The sarcoplasmic reticulum, terminal cisternae around the T-tubule. So now let's go from a fiber to a fibril to now the myofilaments. So we already discussed what a fibril looks like. It is a rod of proteins. Well, what makes up this rod of proteins? So up here is our myofibril, one rod, and now we're slipping out myofilaments. The myofilaments are called thick or thin filaments. And a single thick filament, a single myosin molecule, looks like this. It's 2 proteins that are twisted around each other, and it has 2 heads. I'm going to be drawing this on the board for you, and the language of muscle contraction sounds very dirty, but it is officially the language of muscle contraction. I promise you. I promise you. I'm not trying to be crude or dirty, but I have to use words like cocked, power stroking, slumping. It all sounds very suggestive, but those are real terms that are used to describe muscle contraction. Myosin has heads that can bind to actin. Actin is shown here in these red ball-like structures. Actin is part of a thin filament. It is a globular protein, and it looks like it's a double-stranded pearl necklace that's twisted around itself. Shown here in green is another part of a thin filament, and that is tropomyosin. Tropomyosin is also looks like a more delicate, smaller pearl stranded necklace, double-stranded, twisted around itself, and twisted even more around the actin. Shown here in blue are the troponin proteins. They are not filaments. That's what I was trying to say. There are 3 subunits to it. One subunit is anchored to the actin. One subunit is anchored to the tropomyosin, and a third subunit is going to bind to calcium when it is released from the sarcoplasmic reticulum. Single molecule myosin, but really when we make a thick filament, we have a whole bunch of them bundled together. It's sort of like picking a whole bunch of flowers and bundling them together, and those flowers kind of go around in every direction. Or you can think of it as a golf club, a whole bunch of them in a golf club bag, and their heads just kind of go all which way. But it is two-headed. There are two heads. And when we get into the muscle contraction after our break, I am actually going to make a reference to these structures to a TV show that was called All in the Family. And most of you have not heard of that program. You do, don't you? All in the Family, back in my day, was as edgy and socially questionable as Family Guy is. And if you think about the theme song for Family Guy, it seems today that all you see, right? And they're at the piano. Well, the opening theme song for All in the Family is Archie and Edith sitting at the piano, and they're singing Those Were the Days. And again, it pushed the boundaries as much as Family Guy does and as much as South Park. Lots of the references in Family Guy are actually derived from All in the Family. So after our break, I'm going to show you some clips from that TV show so that you get a visual of the characters that I'm going to be telling you about. Okay, a thin filament is consisting of three structures. You have the actin protein, double-stranded globular protein twisted around like a double-strand pearl necklace. Tropomyosin, again, globular, two strands twisted around, and then also twisted around actin. And then we have troponin. And troponin has three globular subunits, one anchored to actin, one anchored to tropomyosin, and one that will bind to calcium when the sarcoplasmic reticulum lets it free. When we look, I hope this is review. I hope you learned about A bands and I bands. Does any of that ring a bell? A sarcomere, the functional unit of a muscle cell. The sarcomere is the functional unit of skeletal muscle. And it is bounded, one sarcomere is bounded on each side by a Z disc. And a Z disc is a series of proteins that sort of act like an anchor that a boat would use when they want to be stable, right? And not just drift through the ocean. If we look at this under a microscope, we see that there are some patterns. For example, it looks like this is very light, isn't it? A lot of the light from the microscope is getting through this tissue specimen. This is called the I band, and it straddles across a Z disc. It is part of this sarcomere, and it is part of that sarcomere. There's a reason why it's called I band. That's because it's called isotropic, and you don't need to worry about that word. Here's my trick. What is the second letter in the word light? I. So look for the light part, that's an I band. Then over here we have an M line, and the M line is for middle. M for middle. It's the middle of the sarcomere. And then we have an A band. And an A band looks very dark, and this is where we have actin and myosin overlapping each other, like this. And because they're overlapping, the light does not get through as well. And when you're looking at this specimen under the microscope, your eye perceives it as a shadow. Again, because it's blocking the light coming up from the light source. It is called anisotropic. You don't need to worry about why. Here's my trick. What's the second letter in the word dark? A. So where you see a lot of dark, that's the A band. Now over here, you see where it's not as light as the I band, but it seems a little light when we get close to the M line. Can you see that? That is where we only have thick filaments. We don't have overlap of the thin. That's why some more light is coming through from the light source and hits the retina of our eye, and we see, okay, so this looks dark, and this looks light, and this looks a little bit lighter on either side of the M line. Again, only from thick filaments. So you can see that here. On either side of the M line, we only see thick filaments. What's the second letter in the word thick? H. Thick filaments. Only thick filaments. So we learned about our I band and A band, M line, H zone, Z disc. And this is all from the regular arrangements of myofilaments. This is a classic picture of skeletal muscle. Okay, you need to know when the muscle contracts, when the sarcomere shortens, the myosin is going to pull on the actin filaments, much like people playing tug of war. And when you play tug of war, have you ever played that? You have a team, there's a team on the other side, there's a little flag in the middle, and the idea is to get the flag to cross over towards your side. Okay, when you play tug of war, do you and your teammates just go, okay, everyone let go, and then grab forward? I mean, that's hilarious when you let go all at once. What's going to happen to the other team? They win by default. Of course they do. But if you're really competitive and you want to give them a good fight for winning, you don't just all go at the same time. That would loosen the rope, and then they would win. What do you do instead? It's hand over hand, and different parts of your team are going to be reaching forward and others are going to be pulling back, right? So that's what myosin heads will do as well. They're going to attach to actin and pivot and pull the actin, the thin filaments, closer to the M line, And when the sarcomere is fully contracted, and don't worry, we're going to watch some videos on this. When it is fully contracted, the I and the H, the I band and the H zone disappear. How can you remember that? Hi, bye. They disappear. Okay, let's review what happens, how a neuron fires. And I'm going to tell you this. If you want to videotape me, if you want to record me, feel free. I am recording it here. I'm going to do my best to make sure I am in the line of sight for this video. But if you want your own personal video, feel free. Skeletal muscle is innervated by a somatic motor neuron. That means soma means of the body. And this motor neuron starts from the ventral horn of the spinal cord and travels through the spinal nerves, etc., to get to the skeletal muscle. And we'll talk about motor units in a little while. Right now, I just want you to think about a one-to-one interaction, okay, between this neuron and the skeletal muscle cell. Our story actually begins when the neuron cell body, the dendrites, bind a neurotransmitter that was released from another neuron in the spinal cord. And that somatic motor neuron becomes less negative, more positive. It is depolarizing. It is experiencing an EPSP. It is getting closer to threshold. As that local potential, graded potential, ripples through the dendrites to the soma, it will reach the axon hillock. This is where the all or none decision is made. If the cell depolarizes enough, which voltage-gated channel will open first for the most electrical current? The dance of the gates. Which one do you want to open first? We went from minus 70 to roughly minus 55. Which gate, which voltage-gated channel should open? Sodium. And then remember me running, and the cell has an action potential, which means a temporary reversal of membrane potential. Becomes very positive, close, but does not get to sodium's Nernst potential. And then the potassium voltage-gated channels open. The cell, the neuron, hyperpolarizes for just a little bit, and then the sodium-potassium ATPase starts to clean up the mess. Well, this action potential ripples down the axon. Remember when we looked at a myelinated versus a non-myelinated axon? It's much faster. We talked about saltatory conduction. Well, when this action potential reaches the terminal bouton, synaptic terminus, axonal terminus, there's always multiple names, it opens calcium voltage-gated channels. And calcium voltage-gated channels open and calcium diffuses into the terminal bouton. This causes synaptic vesicles to fuse and exocytose into the synaptic cleft. And that neurotransmitter that is released is acetylcholine. And the acetylcholine diffuses across that synaptic cleft, part of the neuromuscular junction, and it will bind to receptors on the skeletal muscle sarcolemma. The most abundant one is the nicotinic acetylcholine receptor, but there is also some muscarinic acetylcholine receptors. This acetylcholine is going to allow a lot of sodium to come into the skeletal muscle cell, depolarizing it. This is called a motor end plate potential. So the skeletal muscle now is going to depolarize, reach threshold, sodium voltage-gated channels start opening all along the sarcolemma and down the T-tubules. And right next to the T-tubules, I'll remind you, is the terminal cisternae of the sarcoplasmic reticulum. And as that action potential goes down the T-tubules, it triggers the opening of calcium voltage-gated channels. And here comes calcium, and it comes out. So up until the point of releasing calcium, the neuron releases acetylcholine, then the skeletal muscle cell has a motor end plate and spikes its action potential. That is all called excitation. All of that is excitation. Okay? So you can stop recording now, because I'm going to move on. And yes, muscle does have potassium voltage-gated channels, just like neurons do, to help reset. The action potential has to go back toward our starting point. There are 5 components to this communication between the neuron, somatic motor neuron, and the skeletal muscle cell. This is called a neuromuscular junction. The 5 parts are: 1, the cell membrane of the neuron. It's called a neurolemma. Again, lemma, plasma lemma means cell membrane, neurolemma. 2, the neurotransmitters that are found in the vesicles. So it's the vesicles filled with neurotransmitter. 3, the actual cleft, the gap between the neuron and the skeletal muscle membrane. 4, we have receptors on the sarcolemma, that means on the skeletal muscle membrane, most of which are nicotinic acetylcholine receptors. Most. Are there muscarinic? Good. Yes, there are. And 5, we also have the actual sarcolemma. Okay, so neurolemma, synaptic vesicles with neurotransmitter, synaptic cleft, sarcolemma with the receptors that are embedded within it. Now, if we want to, we can give another term for the sarcolemma plus the receptors. If we group them together, that is now called a motor end plate. Okay? So a motor end plate is the sarcolemma with the receptors in it. Some textbooks say that there are 4 parts of a neuromuscular junction, and that's because they're combining 2 together, and they just say motor end plate. The 2 that combine to form the motor end plate is the sarcolemma, the cell membrane of the muscle cell, and the receptors that are embedded in it. And they are ligand-gated receptors. Remember, that's muscarinic, very few of them, but still ligand-gated through second messengers, and nicotinic acetylcholine, where the ligand binds to the actual receptor that has the tunnel going through it, the channel, right? Okay. Now, it might be a little bit hard to see here, but they are describing a motor end plate down here. Motor end plate, they are describing the sarcolemma with these receptors in them. So I just went through excitation with you. I'm going to show you a video right now, and then we'll go through excitation, contraction, coupling, and then contraction. We're going to do it step by step. So I'm going to pause my video, and it's going to take me. So again, these videos that I'm going to show you are under Lecture Unit 2, but you have to download the zip file, open up the PowerPoint, and then slide by slide, click on the link. That's what I'm going to do with you, and also videotape it here. This is all from Pearson Publishing Company.
Skeletal muscle fibers contract as a result of electrical impulses from nerve cells called motor neurons. But how does that cause a muscle contraction? To learn about this, we will divide the process into 3 phases. 1. Muscle excitation at the neuromuscular junction. 2. Excitation contraction coupling. And 3. Cross-bridge cycling. In this module, you will learn about muscle excitation at the neuromuscular junction. Anytime you move your body, your brain or spinal cord sends electrical impulses called action potentials along the axon of a motor neuron to the muscle fiber. The site where the action potential excites the muscle fiber is called the neuromuscular junction.
Sorry, this is the part that takes me a while to navigate. Here we go. 2 parts of the neuromuscular junction.
Let's look at the structure of the neuromuscular junction before we examine how it works. The neuromuscular junction includes the enlarged distal ends of the axon, called axon terminals, and the junctional folds of the muscle fiber. The junctional folds are the region of the muscle fiber membrane, or sarcolemma, with specialized structures for receiving signals from a motor neuron. Let's look more closely at a single axon terminal and the adjacent junctional folds. Notice that the axon terminal and the junctional folds do not touch. They are separated by a gap called the synaptic cleft. The membrane of the axon terminal contains voltage-gated calcium channels. These channels will open in response to an action potential, allowing calcium ions to diffuse into the cell. Within the axon terminal are membrane-bound synaptic vesicles that contain the neurotransmitter acetylcholine. Calcium ions will cause these vesicles to release acetylcholine into the synaptic cleft through exocytosis. On the other side of the synaptic cleft, the junctional folds contain acetylcholine receptors. The acetylcholine receptors are chemically gated ion channels that open when they bind to their ligand, acetylcholine. Outside the junctional folds, the sarcolemma contains voltage-gated sodium channels. Once the muscle fiber is stimulated, these channels open, initiating an action potential on the sarcolemma. This action potential will trigger muscle contraction.
Electrical events precede what? Mechanical, right? You cannot use your laptops, your phones, your computers unless they're charged. You have to charge them to use them.
Now that you know the components of the neuromuscular junction, let's examine how a nerve impulse can stimulate an end plate potential on the muscle fiber to trigger contraction. This process takes place in 6 sequential steps. Right now, our focus is on the first 3 steps leading to neurotransmitter release. In the first step, the action potential arrives at the axon terminal. To understand this step, let us look briefly at how motor neurons work. When the neuron is at rest, the inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane. We call this voltage the resting membrane potential. The action potential that travels along the axon causes positive sodium ions to enter the cell and briefly make the membrane potential more positive. This change in the membrane potential to a more positive value is called depolarization. In the case of an action potential, the membrane charges actually reverse, so that the outside of the axon is briefly negative and the inside is briefly positive. In step 2, the action potential causes the calcium channels to open, allowing calcium ions to enter the cell. Specifically, when the action potential arrives at the axon terminal, the change in the membrane potential opens the voltage-gated calcium channels. Calcium ions diffuse into the axon terminal through the open channels because there is a higher concentration of calcium ions outside than inside the membrane. In step 3, synaptic vesicles release acetylcholine into the synaptic cleft. The influx of calcium ions during step 2 causes synaptic vesicles adjacent to the membrane to fuse with the plasma membrane, releasing acetylcholine into the synaptic cleft by exocytosis.
Getting better at this.
To understand how acetylcholine activates the muscle fiber, we need to look at step 4, in which acetylcholine binds to acetylcholine receptors. Acetylcholine molecules released from the axon terminal diffuse across the synaptic cleft and bind to acetylcholine receptors on the junctional folds. These receptors are chemically gated ion channels that open when acetylcholine binds to them, allowing sodium and potassium ions to diffuse across the membrane. In step 5, the diffusion of sodium ions into the muscle fiber depolarizes the junctional folds. This depolarization is called an end plate potential and represents a small voltage change that only occurs at the junctional folds. Let's look at how this works. Sodium ions are present in a much higher concentration outside the muscle fiber than inside. Potassium ions have a higher concentration inside than outside. In fact, the distribution of these and other ions creates a resting membrane potential, or voltage, across the sarcolemma similar to that of the axon, with the inner surface of the membrane negatively charged. When acetylcholine receptors open, large numbers of sodium ions rapidly diffuse into the muscle fiber, while a smaller number of potassium ions diffuse out. The greater diffusion of sodium ions into the muscle fiber than potassium ions out makes the inner surface of the junctional folds less negative, meaning that the junctional folds are partially depolarized.
At any given time, a membrane potential is a reflection of, oh man, I'm getting scared for you. It is a reflection of the ion with the greatest permeability. And if you paid attention, she said more sodium ions come in than potassium leaves. Sodium had the greater permeability. That's why we're going to depolarize the skeletal muscle cell.
We call this depolarization an end plate potential. Finally, in step 6, the depolarization of the junctional folds triggers an action potential on the adjacent sarcolemma that spreads outwards from the neuromuscular junction in all directions. Remember that the sarcolemma outside of the neuromuscular junction contains voltage-gated sodium channels. When the depolarization of the junctional folds reaches the threshold voltage for the sodium channels on the sarcolemma adjacent to the neuromuscular junction, those channels open, allowing sodium ions to enter the muscle fiber. The diffusion of sodium ions initiates an action potential, which spreads outwards from the neuromuscular junction in all directions along the sarcolemma. You have learned how the activity of acetylcholine at the neuromuscular junction stimulates an action potential along the adjacent sarcolemma. But what causes this electrical signal for muscle contraction to stop? Once signals along the motor neuron cease, additional acetylcholine will no longer be released into the synaptic cleft at the neuromuscular junction. After a brief period of time, acetylcholine that was already present at the neuromuscular junction diffuses away from its receptor site, and the ion channels close. Now, sodium ions can no longer diffuse across the junctional folds. This ends depolarization of the junctional folds and prevents the generation of additional action potentials along the sarcolemma. To prevent re-stimulation of the junctional folds, the acetylcholine in the synaptic cleft must be eliminated. Most of it is broken down by the enzyme acetylcholinesterase, which is located in the synaptic cleft. However, some acetylcholine just diffuses away from the synaptic cleft. With the chemically gated channels closed and acetylcholine removed, the signal for muscle contraction stops.
When we get to the relaxation part, one of the steps for relaxation means, as she said, the neuron needs to stop having an action potential. No more synaptic vesicles should be releasing acetylcholine, and the acetylcholine that was dumped needs to either diffuse away or most of it is destroyed by acetylcholinesterase. And we did talk about things that will inhibit acetylcholinesterase like sarin gas, DFPs, and neostigmines. Neostigmines are a temporary inhibitor. I love these videos. They get it spot on. They really do.
Let's review what you've learned. Each skeletal muscle fiber is stimulated by a motor neuron at the neuromuscular junction. An action potential arriving at the axon terminal opens voltage-gated calcium channels, allowing calcium ions to diffuse into the axon terminal. The calcium ions cause synaptic vesicles to release acetylcholine into the synaptic cleft by exocytosis. Acetylcholine binds to acetylcholine receptors on the junctional folds, opening these chemically gated ion channels. Sodium ions enter the muscle fiber through the open channels, generating an end plate potential. The end plate potential triggers the opening of voltage-gated sodium channels on the adjacent sarcolemma, resulting in an action potential which propagates in all directions across the sarcolemma of the muscle fiber, causing muscle contraction. The signal for muscle contraction stops when the concentration of acetylcholine in the synaptic cleft decreases. Most acetylcholine is broken down by acetylcholinesterase. Some diffuses away from the cleft.
How was that? Helpful? Okay. Let me close these windows. I would definitely look at these videos again that I'm showing you. So let me give you, again, you don't have to videotape me again, I'm videotaping myself. Excitation begins with a somatic motor neuron depolarizing. If it depolarizes enough, sodium voltage gated channels, which are first inserted at the axon hillock, open, and they open all the way down the axon, one by one. We see this upstroke of the action potential, and right behind it we have potassium voltage gated channels that are starting to open, and they help repolarize this somatic motor neuron. If you look at this video again, you'll see that as they showed you the action potential coming down the axon, behind it they showed you repolarization. They did. So this action potential reaches the terminal bouton, the axonal terminus. It causes calcium voltage gated channels to open. Calcium diffuses into the neuron terminal bouton, axonal terminus. This causes the synaptic vesicles to fuse and exocytose the acetylcholine, which then diffuses across the synaptic cleft. This communication between the somatic motor neuron and the skeletal muscle cell is called a neuromuscular junction. There are 5 components to it. We have the neurolemma, we have the acetylcholine in the synaptic vesicles, we have the synaptic cleft, we have the sarcolemma, the cell membrane of the muscle cell, and we have the acetylcholine receptors. Are there 2 kinds there? Yes. Which one's the most abundant one? Nicotinic. Nicotinic. Okay. So now, as the video showed you, these ligand-gated channels are going to open, both the nicotinic and the muscarinic, but muscarinic very few. And that motor end plate potential, as it showed you, is going to spread along the sarcolemma. And once we're out of this invagination where the neuromuscular junction is, you saw the sodium voltage gated channels along the sarcolemma. And she said that this motor end plate is going to trigger those sodium voltage gated channels to open. And it's going to spread in both directions. All of this is part of excitation. There's a little part of it that we're also going to include in excitation. Okay, so we haven't even got to Archie and Edith and Mike and Gloria yet. We're not there yet. But let's watch the next part. I highly recommend you watch this again and again and take notes on what occurs during excitation, contraction, coupling, contraction, and relaxation. Do you understand me? Okay. I don't mean to sound mean. I'm just trying to impress upon you this important stuff. So that was muscle excitation at the neuromuscular junction. Now I'm going to show you excitation, contraction, coupling. So don't be confused. Here we go.
Skeletal muscle fibers contract as a result of electrical impulses from nerve cells called motor neurons. To understand how muscle contraction works, we divide the entire process into 3 phases. The first phase is muscle excitation at the neuromuscular junction. This is covered in the IP 2.0 topic, muscle excitation at the neuromuscular junction.
We just watched
The
this.
second phase is excitation-contraction coupling, the focus of this topic. And the third phase is cross-bridge cycling, which is covered in the IP 2.0 topic, cross-bridge cycling. Remember that muscle excitation at the neuromuscular junction ends by initiating an action potential on the sarcolemma of the muscle fiber. Excitation-contraction coupling is the sequence of events by which the muscle fiber action potential, the excitation, leads to muscle fiber contraction.
Okay, so we're going to modify that answer a little bit so that you have clear boundaries, because otherwise it gets blurred. We're going to watch this, and I want you to see the yellow. Do you see the yellow? That's the T-tubules. Do you see how the side of the sarcolemma is invaginating and going around each of the myofibrils? That's my gum chewing, bubble blowing analogy. It's not breaking through into the cytoplasm. It's just worming its way around. I like that picture. I think it's very helpful.
First, we will examine the structures of a muscle fiber that are responsible for excitation-contraction coupling. These structures include the sarcolemma, the transverse tubules, the sarcoplasmic reticulum with terminal cisterns, and the sarcomeres of each myofibril. Let's begin with the sarcolemma, the cell membrane surrounding the muscle fiber. As we saw in muscle excitation at the neuromuscular junction, a somatic motor neuron stimulates a muscle fiber at the junctional folds, initiating an action potential on the adjacent sarcolemma. Voltage-gated sodium channels in the sarcolemma conduct the action potential over the whole muscle fiber. The transverse tubules, or T-tubules, are regularly spaced tube-like infoldings of the sarcolemma. They branch within the muscle fiber to reach each myofibril. Since they are infoldings of the sarcolemma, the T-tubules also contain voltage-gated sodium channels. When these channels open, they help conduct the action potential from the muscle fiber surface down into the muscle fiber interior. Adjacent to the T-tubules are sac-like bulges called terminal cisterns of the sarcoplasmic reticulum. The sarcoplasmic reticulum, or SR, extends outwards from the T-tubules, forming a web-like network surrounding each myofibril. The membranes of the T-tubule contain voltage-gated proteins. These proteins link to calcium release channels in the terminal cisterns. Together, the voltage-gated proteins and the calcium release channels control the release of calcium into the sarcoplasm. The calcium ions from the sarcoplasmic reticulum activate the sarcomeres of each myofibril. A sarcomere consists of alternating thick and thin filaments. The thick filaments are made of the contractile protein myosin. The thin filaments consist primarily of the contractile protein actin. The actin of the thin filament has binding sites to which myosin attaches during muscle contraction. Two regulatory proteins, tropomyosin and troponin, are also associated with the thin filaments. Tropomyosin blocks the binding sites on actin when the muscle is not stimulated. Troponin holds tropomyosin in place over the binding sites. Calcium binding to troponin causes tropomyosin to shift away from the binding sites.
Again, I'm going to massage this story a little bit as I continue through the PowerPoint. I don't know if you noticed, but did you see that these voltage gated channels, they correctly showed you that there are two gates? Remember the external gate and the internal gate? Absolute refractory, relative refractory, that talk that we had. What? Okay, so we'll go on to the next one. They did explain that the action potential is going to cause the release of calcium through these calcium voltage gated channels. They said a voltage gated regulated protein and then these calcium release channels. Just group it together as calcium voltage gated channels.
The key to excitation-contraction coupling is an increase in calcium ions within the sarcoplasm. We just learned what causes the release of calcium ions from the terminal cisterns of the SR. Now let's see how the calcium ions allow the sarcomeres to contract. To understand the role calcium ions play in ECC, we will need to look more closely at the thin filaments. Each actin has a binding site for the myosin heads, but in the absence of calcium ions, these sites are covered by tropomyosin. Calcium ions released from the terminal cisterns bind to troponin. This causes troponin to change shape in a way that shifts tropomyosin away from the myosin binding sites on actin. Once the binding sites are exposed, the myosin heads can bind to them, forming cross bridges between the thick and thin filaments. Muscle contraction can now occur through cross-bridge cycling. This process is covered in the IP 2.0 topic, cross-bridge cycling.
Did you see how the I zone and H zone and I band shrank all the way? Yeah. When she says ECC, just so you know, it means excitation contraction coupling.
Let's review what we have learned. Stimulation at the neuromuscular junction triggers action potentials that spread across the sarcolemma and down the T-tubules. In the T-tubules, the action potentials cause voltage-gated proteins to undergo a shape change that opens calcium release channels of the terminal cisterns. Calcium ions released from the terminal cisterns bind to troponin, causing a shape change that pulls tropomyosin away from the myosin binding sites on actin. Myosin heads can now bind to actin, and the muscle fiber contraction can occur. As a result, calcium ions are responsible for coupling electrical excitation to the contraction of skeletal muscle fibers. Skeletal muscle fibers contract as a result of electrical impulses from nerve cells called motor neurons. To understand how this happens, we divide the entire process into 3 phases. First, the neuron stimulates the muscle fiber. This is covered in the IP 2.0 topic, Muscle Excitation at the Neuromuscular Junction. Second, the muscle fiber action potential leads to muscle fiber contraction. This process is explained in the IP 2.0 topic, Excitation-Contraction Coupling. Third, muscle contraction occurs through cross-bridge cycling, which is the focus of this topic. Remember that excitation-contraction coupling triggers calcium ions binding to troponin, causing the myosin binding sites on actin to become uncovered. Cross-bridge cycling is the sequence of events where the myosin of the thick filaments pulls on actin, which slides the thin filaments toward the center of the sarcomere and results in the shortening of the sarcomere. Multiplied over all the...
Did you see the H zone and the I band shrink? Make sure you watch that again.
The sarcomeres in the myofibrils, this results in muscle contraction. To start, let's magnify a myofibril and examine the features of the sarcomere. To start, let's magnify a myofibril and examine the features of the sarcomere.
That was spectacular.
Now that you know the regions of the sarcomere, let's examine the sliding filament mechanism of muscle contraction. Recall that the thin filaments extend from the Z-disc toward the center of the sarcomere, overlapping partially with the thick filaments. According to the sliding filament mechanism, a muscle contracts because the thin filaments slide past the thick filaments toward the center of the sarcomere from both sides. In doing so, they pull the Z-disc at either end of the sarcomere toward the central M line, shortening the sarcomere.
H zone gone.
This occurs at all the sarcomeres along the length of the muscle fiber. Let's look at how the bands change during muscle contraction. Notice that the I band shortens as the sarcomere goes from a relaxed to a contracted state. However, the width of the A band remains the same length. This is because the filaments themselves do not change in length, they just overlap more. Now let's focus on the H zone. Like the I band, the H zone narrows and may even disappear as the thick and thin filaments overlap more and more. How does this sliding movement occur? During muscle contraction, when myosin heads bind to actin, they form cross bridges. Once cross bridges form, myosin pulls the thin filaments toward the center of the sarcomere. The pulling motion of the myosin heads is similar to how the oars of a rowboat work. The thick filament is the boat, and the oars are the myosin heads that form cross bridges. Each stroke produces only a little movement. However, just as repeated oar strokes move a boat, so the repeated breaking and reforming of myosin cross bridges pulls actin toward the M line.
It looks like tug of war, doesn't it? Do you see the heads taking turns? And I don't know if you noticed this part. But this myosin thick filament has 2 hinge points. There's actually this hinge point, and also the heads can pivot as well. And I'm going to explain that to you more. They don't really cover it, but you can see the bend here. And then as we watch this, the heads will actually bend.
A little movement. However, just as repeated oar strokes move a boat, so the repeated breaking and reforming of myosin cross bridges pulls actin toward the M line. A single action potential is sufficient to stimulate many cross-bridge cycles. This shortening takes place in all the sarcomeres along the length of each myofibril and in all the myofibrils of a muscle fiber. The result is muscle contraction. Now that we know that thin filaments slide over thick filaments during muscle contraction, let's examine how the process works in detail. Myosin molecules have two important features that enable the thick filament to bind to and move the thin filament. First, the hinged portion of the tail allows the myosin head to reach the binding sites on actin. Second, the myosin head can pivot back and forth between high energy and low energy positions. This pivoting motion constitutes the power stroke that pulls the thin filament along the thick filament. A sarcomere shortens when myosin heads on the thick filaments form cross bridges and pull on the actin molecules in the thin filaments. The process known as the cross-bridge cycle consists of 4 steps. 1. Cross-bridge formation. 2. The power stroke by the myosin head. 3. Cross-bridge detachment. And 4. Reactivation of the myosin head. Let's examine each step. Step 1. Cross-bridge formation. The cycle starts when excitation-contraction coupling causes the myosin binding sites on actin to be exposed. Myosin heads bind to these sites, forming cross bridges. Myosin starts in its high energy state, in which the ATP binding site on the myosin head is occupied by ADP and
high energy state, I'll explain it to you again in a different way. It's like the catapult, the arm of the catapult being pulled down. That takes a lot of energy and a lot of people to do that. That's cocked. They don't like using the word cocked anymore, but I can show you a textbook where they use that word. High energy state, that catapult arm is cocked. C-O-C-K-E-D.
phosphate. The myosin head is ready to pivot for the power stroke. Step 2 is the power stroke. Release of inorganic phosphate from the myosin head causes the head to pivot, pulling the thin filament toward the center of the sarcomere. ADP is then released as well. The myosin head is now in its low energy state, but it remains attached to actin. The energy for the power stroke is ultimately provided by the ATP that binds to the myosin head. Step 3 is cross-bridge detachment. Myosin remains attached to actin until an ATP molecule binds to the empty ATP binding site on the myosin head. This weakens the link between the myosin head and actin, and the myosin head detaches. The detached myosin head remains in its low energy state. Step 4 is the reactivation of the myosin head. This occurs when the ATP bound to the myosin head undergoes hydrolysis to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, pivoting it back to its high energy state. The cross-bridge cycle starts again when the energized myosin head forms another cross-bridge. As long as the myosin binding sites on actin remain exposed and ATP is available, the cross-bridge cycle repeats, pulling the thin filaments toward the center of the sarcomere. During muscle contraction, cross-bridge cycles occur between the thick and thin filaments along the entire length of each sarcomere. But not all myosin heads are bound or disconnected at the same time. They can be in different steps of the cross-bridge cycle. Remember that there are thousands of sarcomeres in a myofibril and thousands of myofibrils in each muscle fiber. Cross-bridge cycling at all of these sarcomeres causes the entire muscle to contract.
Relaxation.
Cross-bridge cycling ends when the concentration of calcium ions in the sarcoplasm decreases to the resting level of the unstimulated muscle. Two events are responsible for this decrease in calcium concentration. First, the terminal cisterns cease releasing calcium ions. This occurs when signals from the motor neurons stop and no additional action potentials conduct along the sarcolemma and T-tubules. Second, calcium ions are actively transported out of the sarcoplasm back into the sarcoplasmic reticulum by specialized calcium pumps. This calcium transport reduces the calcium ion concentration in the sarcoplasm below the threshold necessary for excitation-contraction coupling. The reduced level of calcium ions causes troponin to return to its original shape, causing tropomyosin to glide over and cover the myosin binding sites on actin. Even though some of the myosin heads are in the high-energy state, they can no longer form cross bridges. The sarcomeres return to their original resting length and the muscle relaxes. Let's review what you've learned. During a muscle contraction, sarcomeres shorten because the thin filaments are pulled past the thick filaments toward the center of the sarcomere. This is called the sliding filament mechanism. The thin filaments are pulled by the myosin heads of the thick filaments, which continuously form and break thousands of cross bridges with the actin molecules of the thin filaments. Cross-bridge cycling is the sequence of events where myosin pulls on actin. The four steps of the cross-bridge cycle are the formation of cross bridges, the power stroke, cross-bridge detachment, and myosin head reactivation. Cross-bridge cycling continues as long as the myosin binding sites on actin are exposed and ATP is available. Muscle relaxation occurs when the calcium ion concentration drops below its threshold value for binding to troponin. This causes tropomyosin to shift back to cover the myosin binding sites on actin. Cross bridges are now prevented from forming, so the thin filaments slide back to their resting position. The sarcomere is now ready for the next stimulation at the neuromuscular junction to trigger another muscle contraction.
Okay, did you find that helpful? So I'm going to do it again. If we look at this picture right here, in purple are the actin thin filaments, the actin component of the thin filament. And again, I want you to think of that as Gloria from that TV sitcom. She's always exposing herself, or wants to. And the light blue, I want you to think of tropomyosin as Archie. He's always trying to cover her up. And the yellow, the three subunits here, I want you to think of Edith, because she was a very sensible one, very attached to her husband, but also very attached to her daughter. And she wanted grandchildren. And she was sensible, just like troponin is. When calcium is around, it binds to that third subunit. And troponin changes position. That's called the conformation change. And it pulls tropomyosin with it. If I can act it out and be Edith, she would say, Archie, come with me. Let's go for a walk. She knew when the newlyweds needed to be alone, and they would go for a walk, and bow, bow, chicka, chicka, bow, bow. What's going to happen? What's going to happen? Power stroking. Okay. Y'all are looking like you're getting tired. I am too. It's been a long week of lecturing. So let me tell you about our divisions for testing purposes. I've gone through excitation. I've told you that the motor end plate potential generates an action potential by opening sodium voltage gated channels and even the video, in all directions and down the T tubules. That action potential is going to open calcium voltage gated channels. That marks the end. That marks the end of our excitation. Now we have excitation contraction coupling. The calcium that is now released diffuses in the sarcoplasm, in the cytoplasm, and it's going to attach to troponin. Troponin, Edith, Archie, come with me, take a walk with me, is going to cause tropomyosin to shift, as you saw in the video, exposing actin's binding sites for myosin. And think M for myosin and M for Michael. He is always cocked. It's like the catapult being pulled back. It took energy to do that. ATP had to bind and be hydrolyzed into ADP, an inorganic phosphate. Myosin is always cocked and ready to go. As soon as actin is exposed, that ends our ECC. Once actin is exposed, now we can have power stroking occur. And as they showed you, myosin has a hinge point. When actin is exposed, myosin hinge point brings the heads to bind to the actin binding, the myosin binding sites on actin. Then we have power stroking. The heads pull on the thin filaments. Inorganic phosphate moves away, is detached. The myosin heads cannot let go until a fresh ATP binds. When the fresh ATP binds, takes over the place of where the ADP was, then the myosin heads detach, and when that ATP is hydrolyzed, we cock again. Catapult is set to go. As long as actin is exposed, we can have the myosin heads bind, power stroke, there goes inorganic phosphate, a new ATP will come and replace the ADP. We then have detachment and then cocking again after the hydrolysis of ATP. In order to have muscle relaxation, they did correctly tell you that we need removal of ACH by acetylcholinesterase. We need the neuron to stop firing an action potential and stop releasing acetylcholine. We need ATP because if we don't have ATP, the myosin heads can't detach. They can't. In fact, this is part of the reason why a person, when they die, they get rigor mortis, because there's no more ATP and the myosin heads can't detach. And those thin filaments are being pulled. After a while, the decomposition occurs, the decomp, right? And the proteins are destroyed by proteases, and then the person does not have rigor mortis. And this is how pathologists can try and figure out the time of death. Shortly after death, flaccid, but then we have the dumping of calcium, and myosin is always cocked and ready to go. As the sarcoplasmic reticulum is being destroyed and the calcium comes out, myosin heads with their hinge point, because they've already been cocked, they pull, and the person has rigor mortis. Given enough time, those proteins are destroyed and the rigor mortis disappears. We also need the calcium to be removed from the cytoplasm and back into the sarcoplasmic reticulum. That is called resequestering. We're sequestering the calcium back again. This requires a calcium ATPase. It requires energy. Motor units. A motor unit is a single neuron and all of the muscle fibers it innervates. We can see this motor neuron is communicating to this muscle cell, this one, and this one, but not these other three. A different neuron will have to communicate with them. There are other teachers that teach human anatomy here, right? One, and many of you had you had one teacher or the other one. Okay, they're both wonderful. But if you had one of the teachers, that was your motor neuron. You were the motor unit. That teacher told you what to do, when your exams were, etc. The other half of you had a different teacher. You were that person's motor unit. The teacher, the professor, was the somatic motor neuron, communicating to its motor fibers. That means that you were one motor unit and another. A single muscle has a lot of motor units. Many neurons have to innervate many muscle cells on a muscle, a whole muscle, and act in coordination, hopefully, to get muscle contraction. Next week, when I talk about your PhysioX number 2, they explore motor units again, and I will finesse this a little bit more. When we recruit a lot of motor units, we can get a maximum contraction. Again, I highly recommend that you do PhysioX number 2 to be ready for that conversation. It does a beautiful job showing you motor unit recruitment. Just very quickly, there are different types of muscle fibers. We think of slow twitch and fast twitch, and then there's also intermediate. When we're talking about slow twitch means that they don't have a rapid rate of the cross-bridge cycling. These are usually your postural muscles, muscles that can help you sit upright. They are also considered red fibers, so slow red. And they have a robust blood supply, a huge amount of myoglobin, a lot of ATPase, and their muscle size, the size of those fibers is a little bit smaller compared to the white fast. Let me explain. There are some people, and just think about triathletes. How do they look to you? Do they look very svelte and slender and sleek? Okay. Now, think about Dwayne Johnson, The Rock. Wrestlers. They're bulky, aren't they? Could you imagine them being triathletes? They don't really look like they had the body frame, do they? In actuality, our muscles have a blend of fast white fibers and slow red. But some people, just by genetics, they might have more of one than the other. And that kind of dictates their athleticism. Triathletes, more slow red. They can go the distance, literally. Bodybuilders, fast white. They bulk up. The fibers are bigger. But they're called fast white because they don't have a robust blood supply. And they have very fast myosin head cycling. but they fatigue very quickly. That's why you see some bodybuilders, they lift up the big weight, whoop, and then they drop it on the floor because that's all they can do, right? Lots of muscle, but very short term. Triathletes, like Forrest Gump, right? Running, and it seems like they can go on forever. Slow, red. I can prove it to you because next week when you take your exam, there will be a written component to it. And you might be frantically writing, and I'll watch you. And as you're frantically writing, your interthenar muscles are mostly fast white. And you will do this. You'll put your pencil or pen down and you're going to go, because they fatigue. And you're like, keep working. They fatigue. Postural muscles, long distance triathletes, slow, red. A reflex arc consists of 5 things, and this page number does not apply to the textbook that we are using. A reflex arc is going to consist of a sensor, and we've done this before, an afferent nerve bringing information up to the brain or spinal cord for integration. Then we have an efferent nerve, and it's going to target an effector, in this case, skeletal muscle. So I want to tell you about some reflexes that your skeletal muscle has. Again, a sensor, that's a receptor, an afferent fiber, integration in the brain or spinal cord, an efferent, outgoing, and I'll remind you, the pronunciation isn't afferent or efferent, but I am going to say it that way because afferent and efferent sound dangerously close to each other. The efferent fiber will terminate on your skeletal muscle. That's the effector, something that will respond to the command. Some reflex arcs are monosynaptic, and I'll explain that. Some are polysynaptic. And again, 5 components to a reflex arc. Some reflex arcs are ipsilateral. That means that the stimulus and the effector are on the same side. Some are contralateral, where the stimulus and the effector are on the opposite side. It is involuntary. It is an automatic response. I wanted to say autonomic. So a monosynaptic reflex arc would be your stretch, your patellar stretch reflex arc. That's when the doctor takes the hammer to your patellar ligament and you have your knee-jerk reaction. That is monosynaptic, meaning one synapse from the afferent nerve communicating with the efferent nerve, one synapse. Your crossed extensor or withdrawal reflex, this is polysynaptic. I'm just briefly going to go through these. Trust me when I tell you, these are questions that you can expect to have on your exam, and I believe this is also in your homework packet. Here we have our stretch reflex. Now, the doctor hits your patellar ligament with a little hammer, and this causes your quadriceps to stretch because the patellar ligament is being pushed inward. So it stretches your quadriceps. What can happen to muscle if it stretches too much? That's right, it can be pulled off the bone. It can. So if a muscle like your quadriceps are pulled too much, what do you think they should do? If they're stretched too much, what should they do? I could prove this to you. Do I have a volunteer to come here, lay on their back, and spread their legs like this up in the air and let me push down on your legs? What are you going to do? What are you going to do? What's your reflex? I start pushing. You feel your groin starting to... What do? You don't hit me. What do? You're going to start contracting back. You're not going to relax and go, okay, and then flop your legs. This is detected by the receptor, a stretch receptor. Think S for stretch and S for spindle. It detects when the muscle is being stretched too much. It sends an action potential through an afferent fiber, which then communicates to the efferent fiber. One synapse right there. And it comes down and stimulates the quadriceps muscle group. They contract. That's how a muscle opposes excessive stretching. Now the issue is, though, if we're going to contract the quadriceps, we need the hamstrings to relax. And in order to make them relax, we have to inhibit GABA and glycine. This somatic motor neuron, shown in purple, innervating the quadriceps. I am telling you that this is a question on your lecture exam. The activated muscle would receive acetylcholine, end plate potential, contraction. The opposing muscle group is going to receive an inhibitory signal way up here that shuts down that somatic motor neuron. It does not reach threshold. It does not spike in action potential, and therefore the skeletal muscle and the hamstrings are not activated. And the biggies for inhibitory neurotransmitters are the GABA and glycine. We went over glycine and GABA on a slide. At least GABA, I remember that. Okay. Your crossed extensor is a polysynaptic reflex. And just to show you this, when I was in graduate school and all by myself in a totally different state, I was in San Antonio, Texas, just to have fun and feel more safe and be active, I took a self-defense class. And the instructor would randomly come up to us and pull us by our forearm and yank us like he was a predator, right? Someone waiting for me to come to my apartment door, fiddle with my keys. I had bushes all around my apartment. Anyone could have hidden there. Anyone could have. So he would pull us, and your instinct is when one of your limbs is being pulled, you want to recoil, and the instinct is also the reflex to push with the opposite side. Another way you can think of this is if you're walking along the beach and you step on a piece of glass with your right foot, do you sit there and grind that glass into your foot even more? No, you pull that leg away, but the opposite leg extends. Crossed extensor. Damage to one, pulling of one, recoil. Push away with the other one. Crossed extensor. One limb will flex, the other limb extends. So the thing is, it's very difficult to mentally override reflexes because they are automatic. So we had to train ourselves to realize that our reflex when we're being pulled is to do this and push like this. But a person attacking you is going to anticipate that. They know that's a natural response. So they're going to be pulling even more to drag you with them. We had to learn that not to pull, but to go with them. And they would be off balance because it's like tug of war and you let go of the rope. That's what it's like. And then they would probably fall or at least stumble a little bit, giving you more time to defend yourself. That's really hard to overcome, that instinct. Really hard. So the spindle, think S for stretch. If the muscle is overly stretched, what should it do as compensation? Contract. If a muscle is overly contracted, we also need a stimulus targeting this Golgi tendon organ. And the Golgi tendon organ, I want you to think of T for tendon, and I want you to think of tense. A lot of contraction. And if you contract your muscle too intensely, you can also rip it off from the bone. And I'd like to tell you did I tell you the story about Grace? Okay, let me tell you a quick story about Grace, and then I will give you your goodies, okay? So Grace, I love you, Grace, if you're watching this. Grace was another postdoctorate person in the lab that I was working in. And when I first started there at UC Irvine in Dr. Frank LaFurla's lab, Grace was already part of the softball team. And in the Department of Molecular Biology, the different labs would have their teams and they would play softball. Whoever won, or I should say whoever lost, they had to buy all the beer for the winning team. They would all go out and have a good time, right? So beer is on the line. So I'm sitting in the bleachers and I'm sitting with her husband, Manny, who's an emergency room doctor. Grace is playing catcher. Now, I know you can visualize a catcher being in the catcher's position. They're on the balls of their feet. They're squatting and holding that. So Grace is the catcher. The batter comes up, hits the ball. It's the second. It was 2 outs, 1 more out, and beer will have been gained. So pop fly, foul ball. If Grace catches it, game over. Beer. Grace springs into action. She contracts her gastrocnemius muscles so fiercely, like Barbie walking on the balls of her feet. She just springs up, and that Golgi tendon organ was trying to say to her brain, "Ruh-roh, you don't want to do this." So she was very enthusiastic, caught in the moment. She overrode that safety mechanism. This was before phones had cameras. I'm telling you it happened. I'm telling you. My sons would say, unless you have video footage, it didn't happen. I'm telling you it happened. And I'm going to start laughing because it was one of the funniest things I've ever seen. So her gastrocnemius rips off of her calcaneus. Rips full off. And from the stands, I can see that her gastrocnemius is now balled up behind her knee. Behind her, in the popliteal area. Balled up. And this is Grace. All of a sudden, she starts going, And she starts wailing on the poor batter. And she's hitting the batter, she's hitting her, she's hitting her, she's hitting her. And the poor batter, female as well, just happened that way, is backing up, not knowing what's happening. Why am I being attacked? And here's Grace, like, Grace thought that the batter hit her, you know, like Tanya Harding style, hit her and was trying to get her to miss the ball. That is not what she did. What Grace felt was her muscles snapping off the calcaneus. So here she is, and I'm laughing, crying, and I'm going to Maddie, go get your bride. Look what's, and I said, look, and he can see the balled up gastrocnemius. He goes running from the stands out to Grace, who's still trying to whack this poor woman. He tackles her to the ground, and she's like, let me go! Let me get her! And Maddie's like, honey, we have to go to the emergency room right now. Look at your leg. So the next week, she came back to work. In a boot, they had to attach her gastrocnemius back to her calcaneus. Funniest thing it's not funny, but it was funny. Loved it.